So this is lecture 22, and we're going to start on plate 122. So I'm asking you to skip plate 121, which will be covered in lab, structure and growth of the bone. The endocrine pancreas. We've talked about the exocrine function of the pancreas. Here's our little diagram. You'll recognize this diagram from the GI portion. Right at the entrance in the duodenum, from the pylorus into the duodenum, is the pancreatic and bile duct, sphincter of Oddi. We talked about the production of lipases, amylases, proteases, bicarbonate-rich juices. That was the exocrine portion. 98% of the cells of the pancreas are devoted to the production of pancreatic juices. There is only about 2% of those cells that are focused on endocrine function. Named after the anatomist Langerhans, they look like little islets or islands of different cell types, given that name. No idea on the function. We now know that those cells are critically important in the regulation of blood glucose. We talked about calcium regulation, very tight regulation of calcium. Similar for glucose. We need to regulate glucose very tightly. Main hormones that are produced by these cells, insulin and glucagon. The B or beta cells are the site of insulin production. Alpha or A cells, the site of glucagon. And then there is the gamma or delta cells that produce somatostatin. And it is the concert action of all three of these that help regulate glucose. But calcium is important. Stimulus for the release of these compounds are blood glucose levels. And the cells, in fact, the islet cells, respond to blood glucose levels. So elevations of blood glucose stimulate the release of insulin. Low levels of glucose stimulate glucagon. The release of these two is in part mediated through this third hormone, somatostatin. You see that it tends to have inhibitory effects on both to prevent large surges. The idea is that if you take in a lot of sugar and you get a very high dietary glucose load, insulin levels will go way up. Well, you don't want them to go too high. Similarly with glucagon, so somatostatin acts to inhibit both of these. And you'll note that there is an cross-inhibitory response between insulin and glucagon. So elevations of insulin tend to suppress glucagon. That makes for a more rapid response to blood glucose. So you get a big meal. You sit and you pound all those sugars. You got all those Skittles on board. Blood glucose is starting to shoot way up. You want insulin to go up. You want glucagon to go down because insulin acts to suppress blood glucose. Glucagon acts to increase. Action of glucose. We're going to talk about the actions on the next plate. So let's just talk about the mechanism of action. Insulin, glucagon, somatostatin, all examples of protein hormones. The action of these hormones, then, is through a cell-bound receptor that interacts with the hormone, and through G proteins modulate the calcium uptake, or calcium, we've got calcium on the brain, glucose uptake. All right, so actions of glucose, that's plate 123. Insulin is a feeding hormone. So in response to a dietary intake, particularly of glucose, glucose levels go up. They stimulate the release of insulin. B cells, the beta cells, respond by dumping insulin into the blood. Insulin has as its target tissue liver cells, fat cells, and muscle cells. Interestingly, brain cells and nerve cells are relatively insensitive to insulin. And this will become an important player shortly. So what happens in the muscle cell? There are transporters, called GLUT4 transporters, which are insulin sensitive. When insulin binds to these receptors, it opens up the GLUT4 gates and allows glucose to go into the cells. The glucose becomes important in reformation of muscle glycogen. So this is exactly what you want to do in a cell. Oh, my pizza order. You want to regulate, you want to get glucose back into a muscle cell during a meal. You want to replenish the supplies. The movement of glucose out of the blood and into the cell will act to decrease blood glucose levels. So the stimulation for insulin release, elevated blood glucose. In response, glucose moves into cells, it decreases, and that then suppresses insulin response. So this is kind of negative feedback. In the muscle cell, it's converted to glycogen. Excess amounts of sugar are converted to lipids in the muscle cell. Now, the amount of lipid store, the amount of glycogen store, is relatively limited per cell in a muscle cell. That's not the case with adipose cells. These adipose tissue cells, also under insulin-sensitive transport mechanism, will suck up glucose. Glucose is then converted into free fatty acids and glycerol, both in the fat cell and also in the liver cell. So here, the liver cell, under insulin sensitivity, glucose comes in, glycogen formed, lots of fats produced as well, so you're building back up liver glycogen. You're also producing fat stores. So, right, the whole idea, you take in a big meal, you have lots of glucose available, and if there's excessive amounts of glucose, you're going to convert a lot of that to fat. So when you get these, this is a fat-free diet. Right? So how do they get you to eat a fat-free diet? They'll give you lots of carbohydrates, lots of sugar, so it's very tasty. Lipid, frankly, it's tasty. I mean, why? We have evolved to recognize that as a high energy source. We want to have a lot of energy. Lipids, great for that. Now, when you move into modern America, where you have an abundance, you're sitting in front of your keyboard studying for the online exam, eating food, you're not burning as much. So you have this high sugar diet. It's converting it to fat. So it's not a fat-free diet. Even though the concentrations of fats may be low in your intake, you end up converting that to fat. Why? Glycogen stores are limited. We talked about that in the muscle. You just can't put that much glycogen into muscle. You can't put that much glycogen into the liver either. You have very few glycogen stores in fat cells. So what do you do with it? You convert it to lipids. So insulin stimulates glucose uptake. It also stimulates enzymes involved in glycogen synthesis. So we see this down here. Here are glycogen synthesis, also fatty acid synthesis and triglyceride production. The third stimulus is of enzymes involved in protein synthesis. Again, exactly what you want to do, post-absorptive. You've eaten your meal, you've consumed it, you have lots of energy in your gut. You want to get that material into the body. You want to build your glycogen stores. You want to build up your fat depots, and you want to make proteins. So amino acid building of peptides from amino acids under insulin control. Brain cells do not require insulin, nor does the gut. What that means is glucose uptake by metabolizing brain tissue is unaffected by insulin. Otherwise, think about it. You'd only be smart while you were eating. If you had high levels of insulin and insulin was needed to get glucose into your brain cells, you'd only get that glucose when you're eating. When you go into the fasting mode, insulin levels drop. You no longer have dietary glucose. You are now in a glucose losing mode. Well, if your brain cells were dependent on that, you'd be in big trouble. So we see insulin insensitivity in neural tissue, in the gut, because you don't want insulin necessary to absorb glucose, because it's the glucose that's causing the insulin to be secreted. So it wouldn't make sense. Another tissue that tends not to be affected by insulin is cardiac tissue. So while it's important for skeletal muscle, cardiac tissue is less insulin sensitive. Think of the importance of saving your glucose for brain cell, for heart function, and also red cells. Red cells have no mitochondria, so they, in fact, utilize only glucose. So you have a few cells in your body. Now, they're really important cells, but you have a relatively few number of cells that require insulin. What happens when you fast? No longer dietary intake of glucose. This person is thinking about food, but they're in the fasting sense. They are characterized by decreasing levels of glucose. There is always some requirement for glucose metabolism, whether you're thinking, whether your heart is working, whether your red cells are metabolizing. But in addition, all of your other cells, liver cells, fat cells, muscle cells, have a small requirement for glucose utilization. Well, if you're not eating, where is that glucose going to come from? That glucose is going to come from glycogen stores in the muscle and in the liver, which are under the control of the hormone glucagon. So, in response to low or hypoglucose levels, glucagon is released from the alpha cells. Glucagon attaches to receptors which stimulate enzymes that break down the glycogen. The consequence is to produce more glucose. Glycogen also stimulates the production of new glucose, a process that we refer to as gluconeogenesis. And when you take biochemistry, you will probably find out about the pathways involved, the metabolic pathways involved in the genesis or production of new glucose. Many of these enzymes are under the control of the hormone glucagon. Both insulin, glucagon, and somatostatin are examples of peptide hormones. So the mechanism of action is by extracellular receptors and primarily by changes in cyclic AMP. And we talked about cyclic AMP modulating protein kinases, turning on and off proteins. We come back to that with this explanation. Plate 124. One of the best ways of exemplifying all of the actions of insulin is to look and see what happens when you have an insulin deficiency. Insulin is the only hormone that responds to elevations in glucose. We have a number of hormones which in fact increase blood glucose. We refer to those as hyperglycemic hormones. We've talked about some of them. We talked about epinephrine increasing blood glucoses. We've talked about, now just recently, glucagon. And we're going to talk about some more that are all involved in keeping blood glucose levels high. So we see insulin is the only hormone that actually acts to suppress blood glucose. So what happens when you have insulin deficiency, so no insulin? That is a condition referred to as diabetes. And we're going to distinguish diabetes mellitus from diabetes insipidus. Diabetes mellitus, that is the most common form of diabetes. When somebody says, "Are you diabetic?" and there's some, I don't know what the calculation. We see a lot of diabetes in our domestic animals, particularly in cats. And then certainly in humans. It's one of the number one problems in humans is diabetes mellitus. That is insulin insufficiency or non-responsiveness. Diabetes insipidus, in fact, is a lack of ADH. Mellitus means sweet. Diabetes actually means funnel, and it has to do with the idea of funneling fluids out of the body, and that fluid is sweet. So we're going to see what happens. Decreased insulin. Without insulin, your ability to take up glucose by the cells is inhibited. That is going to produce an increase in blood glucose. Right? Now we said the stimulus for insulin release was elevated levels of glucose. Now we're turning the tables. We're saying, well, the cells aren't producing insulin or the cells aren't responding, so insulin levels, in fact, aren't going up in response to a elevation in blood glucose. So we now have a hyperglycemic condition. Hyperglycemia means elevated levels of glucose in the blood. What are the consequences to that? Normally, all of the glucose is reabsorbed in the kidneys. It's an important metabolic commodity. It's very precious. You want to save. We talked about co-transport of sodium and glucose. We want to reabsorb all that glucose. With it comes water. So what happens when you have elevated levels of blood glucose. Those molecules of glucose get filtered at the proximal tubule in the same concentration that they are in the glomerular capillaries. And I'm going to give you some numbers here because it's probably important to recognize that. So we're going to talk about a normal concentration of 100 milligrams per cent. That means in 100 mils of blood, there's 100 milligrams of blood glucose. Old-fashioned term, but very commonly used. We use it in domestic animals because a lot of the glucose meters that are used in domestic animals, same ones that we use in humans, and it is all kind of calibrated to this kind of antiquated system. So it's not part of the SI system, but it's the way we talk about blood glucose concentrations. We tend to regulate blood glucoses very, very tightly. If you're fasting, that should be the level. Now, that'll go up slightly when you take a meal. But in the fasting state, you should be pretty close to 100 milligrams per cent. 110, ooh, it's high. 90, a little low. So in fact, we regulate these levels relatively constantly. That is the concentration that gets filtered in the proximal tubule. Normally, all of that is reabsorbed. Now, what happens when that number goes up? You start exceeding the capacity for glucose transporters in the proximal tubule. Seen the Lucy, right? She's working in the chocolate pies, and they're coming down a conveyor belt. And then what happens is the number of chocolate pies goes up, and her ability to take it off the... Yes, probably before your time. So anyway, you've got... you only have a certain number of transporters that are working to pull the glucose out of the proximal tubule, put it back in the blood. Well, when this number exceeds about 250 milligrams per cent, you've saturated all the transporters. They are unable to keep up with that presentation of glucose. In pronounced diabetes, these numbers may be 500, 600, 700, 800 milligrams per cent. Huge amounts of glucose flowing into the proximal tubules that doesn't get reabsorbed. Well, if it doesn't get reabsorbed, then you have glucose appearing in the urine. So that is a glucoseuria. Normally, right? No glucose, so now you have glucose appearing in the urine, hence the term mellitus or sweet, and diabetes funneling, because these glucose molecules are osmotically active, water follows. Individuals with diabetes are characterized by huge productions, the production of huge amounts of urine, a polyuria. It's due to the osmotic drag of glucose, but it's a water loss, funneling, if you will, of water out. So that was the term diabetes. Because it's glucose, it's mellitus. Well, what happens in diabetes insipidus? Diabetes insipidus, decreased levels of ADH. Clinical signs of diabetes insipidus, a polyuria. Why? Because you're not reabsorbing it. It's not due to glucose pulling it osmotically. So in fact, instead of being sweet urine, the urine is very bland. It's what's called insipid. If someone calls you and says you have an insipid personality, it's not a compliment. It means you're very, very bland. In medical terminology, an insipid urine means it doesn't have any sugar in it. So the distinction between diabetes mellitus and diabetes insipidus is can be made on concentrations of glucose in the urine. All right, so we have a polyuria, excessive water loss. Well, the hypothalamus responds by secreting ADH, which may have some effect. But again, You can only reabsorb water if you're reabsorbing the solutes. And in the case of diabetes mellitus, it's not. The glucose is not being reabsorbed, and it's taking water with it. It doesn't care about ADH. But in addition, the hypothalamus stimulates thirst. This dehydration so we have a polyuria, a glucose or glycosuria, dehydrating, that produces polydipsia, or excessive thirst. We talk about animals presenting in clinics as being PUPD, polyuric polydipsic. Lots of diseases cause that. If it's an older cat that comes in PUPD, I'm thinking diabetes. How would you know if it's polyuric polydipsic? If it's an outdoor cat, you're not likely to see this. If you have a toilet bowl, you're not likely to see this. So sometimes these cats present, they are PUPD, but the owner has no way of knowing that. Now, let's say it's an indoor cat. Sometimes this polyuria will be in inappropriate places. The animal just can't hold it, so there's spotting on the Berber carpet, or excessive amounts of urine appearing in the with pet litter. Polydipsia, there's a bowl that's set out for the cat, continually refilling that. So there may be a better handle on this PUPD, but again, these are clinical manifestations. All right, what else? You're in this carbohydrate-rich environment. You've got lots of blood glucose. It's not getting into the cells. As a consequence, you start breaking down glycogen. You start breaking down fats. You start breaking down proteins. That as far as your body is concerned, you don't have any glucose. Now, it's present in the blood, but it's not getting into the cells. So it's kind of ironic that now what you're doing is you're saying, well, I don't have any glucose. I need to provide energy. I need to provide glucose, so I'm going to start breaking down glycogen. That's going to further exacerbate. What happens to that glycogen? Broken down to glucose, flows into the blood. That is going to re-emphasize the hyperglycemia. Just make it worse. Breaking down proteins. Storage form for proteins, remember, none. It's all tied up in skeletal muscle. So the breakdown of proteins into amino acids comes at a cost. You start muscle wasting. In the Middle Ages, this was referred to as the pissing evil or pissing disease, because what would happen is these individuals would urinate, polyuric, and they would get thinner and thinner and thinner. So in fact, they were undergoing starvation in the face of all of this blood glucose. So diabetic cats characterized by thin, wasting, decreased muscle mass. You start looking at the urine elevation levels of glucose, and a consequence of excessive fat metabolism, excessive amount production of what are called ketone bodies. So again, when you spend time in biochemistry, you talk about the breakdown of fats. Excessive breakdown of fats can lead to the excess production of ketones, things like acetone. These are relatively toxic. Now, if you've had chemistry, right, you've all cleaned glassware with acetone, that kind of sweet organic smell to it. Diabetics can actually have ketone breath. Cat comes in and breathes on you, kind of go, oh, does this smell like cat food? It smells kind of sweet, organic-y. Ketone in the blood is a ketonuria. ketonuria. Sorry, that's ketones in the urine. Ketonemia is ketones in the blood. Excessive levels of ketones in the blood. Ketones are not a great thing. You need a few, but excessive amounts, think about putting ketone, instead of washing your glassware, putting it into your blood. This is a toxin. This can produce vomiting, altered mental status. Excessive levels in the ketone appear in the urine. That's a ketonuria. As a consequence, one of the diagnostic techniques is to measure glucose and ketones in the urine. So you'll get these little diabetes test strips, which you dip into a urine stream or into a pool of urine, and it tests for the presence of glucose and ketones. Normally, no glucose, normally no ketones in the urine. Presence of ketonuria and glucoseuria, high suggestion that this animal has diabetes. All right, so glycosuria, polyuria, polydipsia. What else? Oh, ketoacidosis. The levels of ketones can be so high that it kills you. Not just GI function and mental status, but can produce a coma. Your book also talks about some other untreated elements of diabetes, nerve and vascular damage. You certainly recognize this in humans. Nephropathies due to glucose, blindness, sorbitol deposition in the lenses. So there are a lot of other sequelae to untreated diabetes. Hypertension. Diabetics oftentimes have hypertension, so we see that untreated diabetes mellitus is a big problem. Your notes talk about two types. We recognize both types in our domestic animals, type 1 and type 2. Type 1 is insulin dependent. This tends to occur in young individuals. Does it talk about it? Yeah. So type 1. Insulin dependent tends to be in young at birth. Insulin insufficiency due to a lack of production. So you look at the pancreas, you look at the islets of Langerhans, the beta cells are small, non-existent. You stain, you look for insulin, there's none. So there's been a failure. These individuals then are insulin dependent at birth. The other type 2, is what we call acquired insulin dependency, or non-insulin dependent, because insulin is being produced. The body is not responding to it. This importance of modulation of all of these hormones. If you turn on hormone levels and you keep them high, what happens is the receptors downregulate. They stop production numbers and sensitivity. Fewer receptors, less sensitive, you can have plenty of insulin, but the cells just aren't responding. So it's as if they can't see that insulin. Causes for type 2. One of the big causes for type 2 diabetes is excessive sugar consumption. So it tends to be a disease more of the affluent societies rather than the non-affluent societies. We also see it in domestic animals, what is called an acquired insulin sensitivity. How do you treat it? You give them insulin. In the 1920s, the importance of insulin in regulation of blood glucose and the site of the production of insulin was identified in dogs. Eli Lilly Pharmaceutical Corporation came up with this. And Eli Lilly's in Indiana, big center for the meat market of the bread basket of the United States. A lot of meat slaughtering done there, a lot of pig processing, a lot of bovine processing, and Eli Lilly affiliated with that because they were harvesting pig pancreases, collecting the cells and collecting the insulin from pig pancreases, also from bovine pancreases. Now, the amino acid sequence of those insulin molecules is slightly different than human insulin. So one of the consequences to prolonged insulin therapy is you continually are introducing a protein, a foreign protein, into your body. One of the big concerns about type 2 or type 1 diabetes in particular was this was a lifetime disease. These people were using insulin from a very early age. The only insulin they were able to get was from pigs or from cows. And one of the problems was an immunity against that insulin. So you'd be taking adequate amounts of insulin, but your antibodies would be tying it up before it would have an opportunity to affect the cells. So in fact, you are now back into the diabetic situation. Treatment for that was usually to change the origin. You know, you were on pig, go to bovine. Well, now, with the advent of recombinant DNA, we're able to splice the gene into an E. coli and produce human insulin. So it's called Humulin now, so the compound you're getting now is an exact match. It does not have those side effects, those immunological side effects of using either swine or bovine insulin. And I think we'll stop there.